Hi everyone, welcome back to this short tutorial from Pathology Made Simple at ilovepathology.com. This is the part 2 of Diseases of Immune System series. In this part, let's learn about adaptive immunity. So, initially, to begin with, we are trying to understand the normal immune response, right? So, in the part one, I talked about innate immunity, and we did discuss about differences between innate and adaptive immunity. And in today's session, we will learn more about adaptive immunity. We will look into the types of adaptive immunity, like humoral versus cell-mediated. We will talk about lymphocyte diversity. We will understand in detail about the T and B cells, including their receptor structure. We will talk about dendritic cell, macrophages, and end with understanding the tissues of immune system. So we know that adaptive immunity consists of lymphocytes and their products. When I say products, which means including antibodies. So they are basically categorized into two types: humoral immunity and cell mediated immunity. Humoral immunity means we have protection against extracellular microbes and their toxins, whereas cell mediated immunity is what the defense against intracellular microbes as well as against cancers. Right? So humoral immunity is mediated by B lymphocytes and their products. B lymphocytes, they get converted to plasma cell, and these plasma cells produces antibodies. So B lymphocytes and their products called antibodies, which are also referred to as immunoglobulins, whereas cell-mediated immunity is mediated by T lymphocytes. T for thymus-derived lymphocytes. What you need to know is that both these cells, B lymphocytes as well as T lymphocytes, they have very highly specific receptors for various substances, and these substances are referred to as antigens. So the lymphocytes which are involved in various immune response, they're not fixed. Fixed matlab they're not situated in one place. So they're constantly circulating among lymphoid organs and other tissues via the blood and the lymphatic system. And by doing so, these lymphocytes, they are constantly observing and then looking for various invaders in the form of antigens, and that's what we call it as immune surveillance. These lymphocytes goes to the site of infection and then they act against them. And that's what we call it as homing of these lymphocytes at the site of infections. In the lymphoid organs, the lymphocytes are segregated and then they interact with one another only when they are stimulated. If the lymphocytes have not encountered their specific antigens, then they are referred to as naive cells, whereas the lymphocytes which are activated by the recognition of various antigens, they are called effector cells. The third type of cells are memory cells, and these are lymphocytes which live in a state of heightened awareness. The effector cells are the ones which eliminate the microbes, whereas memory cells, they react rapidly, they react very strongly, and when these microbes return, they remember that there was once this microbe has affected our body. So let's understand a bit about lymphocyte diversity. So we know that lymphocytes are like soldiers. Imagine that your body is like a country. In our body, these lymphocytes are like soldiers, constantly protecting against various invaders. And invaders in our case is mostly microbial pathogens. The antigen these lymphocytes encounter are like enemy to these soldiers. So what we need to realize that all these lymphocytes are trained to recognize all kinds of antigens. Where are they trained? They are trained either in the bone marrow or in the thymus. We will talk about it a bit later. Imagine a situation where the antigen appears. Then what happens? Then group of lymphocytes which are specifically trained to recognize this particular antigen, they come together. Okay, they come together, and this group of lymphocytes are referred to as clone. And this is what we mean by clonal selection. Now, what is clonal selection? It is basically right soldier for right enemy, right group of lymphocytes, a right clone of lymphocytes which can act against that particular antigen. That's what we mean by clonal selection. Now, there are trillions of lymphocytes in our body, and there are millions of clones. And each of these clone is like a specialized team among the soldiers with unique badge. And these badges is what is antigen receptors, which identify a particular antigen. Right now, let us see how our body creates so many type of receptors, so many unique teams, so many clones. How is that our body creates? That means we're looking at antigen receptor diversity. Okay, during the development of these lymphocytes in the thymus and the bone marrow, what happens is that the gene segments of these lymphocytes, they are assembled by recombination and DNA sequence variation. Okay, so the gene segments are assembled by this particular process, and there is mix and match which occur. The shuffling of these DNA sequences, and because of lots and lots of mix and match which occurs during the process of development, it creates lots and lots and lots of different type of genes, and that is the one which creates diverse antigen receptors. Okay, that means during the process of development itself, you are exposed to n number of antigens, and for that there is development of receptor which can recognize those antigens, and these are B and T cell receptors. Because they are exposed to so many kinds of antigens, by that mix and match or shuffling of these DNA sequence and recombination process, they can recognize all forms of antigen later to combat enemies in the body. Right now, what is the significance of knowing this antigen receptor diversity? And first and the foremost thing that we know that it is very very important for the body to fight against infections. That's one thing. Second important significance of having this antigen receptor diversity is that we know that there are different clones of cells. Knowing these clones of cells helps in identifying lymphoid neoplasms. When you suspect a neoplasm, and then do a marker which can identify that particular clone, so it not only helps in fighting infections, it can also be used to diagnose lymphoid neoplasms. Now let's learn about various types of immune cells, which are T lymphocytes, B lymphocytes, macrophages, dendritic cells, and so on. The first one is a T lymphocyte, which are like a special agent in your army. There are three different populations of T lymphocytes. One is called helper T cells. Second is cytotoxic T cell. The third one is regulatory T cell. Helper T cells are like generals, whereas cytotoxic T cells are the ones which are like elite soldiers, whereas regulatory T cells are like peacekeepers. Helper T cells, they are the ones which stimulate B cells to synthesize antibodies. They don't act directly. They stimulate the B cells to synthesize antibodies, and they also activate other leukocytes, for example, phagocytes, to destroy microbes. That's the role of helper T cells. Whereas cytotoxic T cells, as the name says, they are the ones which kill the infected cells, particularly the viral infected cells and also the cancer cells. The third one being regulatory T cells, which prevent reaction against self antigens. Okay, that's very, very important because that is the one which prevents no autoimmunity, and they also limit immune responses because the body knows that too much of inflammation can be harmful. Okay, so that's the role of regulatory T cells. So three major populations: helper T cells, cytotoxic T cells, and regulatory T cells. Let's learn a bit about development of T lymphocytes. These are developed by from the hematopoietic cells in the bone marrow, which moves on to the thymus. Thymus is a school where these lymphocytes mature. They learn to identify self and non-self antigens, and they become mature T cells. And these mature T cells, they move on to the blood, where 60 to 70% of circulating cells, circulating lymphocytes, are T cells, whereas the remaining, move to the secondary lymphoid organs. Let's learn about T cell receptor. That's very, very important to know how T cell functions. Consider this is a plasma membrane of a T cell. So this structure is the T cell receptor, right? TCR stands for T cell receptor. It is made up of two different protein chains. Look at this. This is one protein chain. This is another protein chain which is linked or connected together by disulfide bond. And each of these proteins chains has two parts. One is the part which recognizes the antigen, that is the variable region, whereas the second one is the one which is attached to the cell membrane, that is the constant region. So this T cell receptor is the one which recognizes antigen. And remember, the T cell receptor does not recognize antigen just like that. It needs the help of another cell which is called as antigen presenting cell. And that could be cancer cell as well. Because these antigen presenting cells on their plasma membrane have these molecules which can present the antigen, and this is called as major histocompatibility complex. Okay, that's a molecule presenting on the antigen presenting cells. So this T cell receptor is also helped by other molecules called CD3. Okay, this is another molecule which is like CD3 and the zeta molecule. Okay, so the CD3, zeta, and the T cell receptor together constitutes T cell receptor complex, right? So these molecules augment the antigen recognition process as well as the signal formation. As I told you, the T cell receptor, along with the T cell receptor complex, identifies the antigen only when it is presented by the MHC complex, and this concept is referred to as MHC restriction. So this MHC restriction ensures T cells to identify the target cells which are infected or abnormal. And we should also note that normal cells do not display these MHC peptides. MHC restriction helps in focused and controlled immune response. There are two other molecules which you need to know. One is CD4. Okay, CD4 is like a special access card, that helps the T cells to connect to different types of MHC molecule. CD4 is the one which identifies MHC class 2 molecule. In the next session, I will be discussing in detail about the concepts behind MHC. As of now, just remember that CD4 identifies MHC class 2 molecule, and the cell which has the CD4 is called as helper T cell. Whereas cytotoxic T cell has CD8, which identifies the MHC class 1 molecule. Having understood the concepts behind the first signal, let us see if there are another signal. Let us see if there is another signal involved. That is by the help of CD28. Okay, this CD28 is present on the T cell, which binds to the ligand on the antigen presenting cell, and that ligand is CD80. So this ligand receptor interaction provides second signal for T cell activation. So this is the first signal, the major signal. The second signal is by CD28 and CD80 interaction. This is the second signal, which is also called as co-stimulatory signal. So these are referred to as co-stimulators. See, the concept of understanding signal 1 and signal 2 is that if you have both these signals, it means or it ensures that T cells are properly activated to respond to specific antigens. That's the importance of knowing two kinds of signals. Now let's move on to understand the B lymphocytes. B lymphocytes are the only cells in the body which are capable of producing antibodies. And you know that antibodies are mediators of humoral immunity. So during development, we know that hematopoietic cells in the bone marrow, they mature into B cells within the bone marrow itself. And once they mature, see 20 to 10 to 20% of mature B cells goes to the circulation. Remaining 80 to 90% of them reside in the secondary lymphoid organs. And these are lymph nodes, spleen, and mucosa associated lymphoid tissues. How do B cells work? No, just like T cells, they also have receptors which recognizes antigen through a receptor named B cell receptor. Ultimately, once they recognize antigen, the B cells gets converted into either plasma cells or memory B cells. But these plasma cells, we know that plasma cells are the protein factories for producing antibodies. Hundreds and thousands of antibody molecules are produced per second, whereas memory B cells are the ones which live for a long time. They remember the specific pathogen. and then they respond much more quicker if these pathogens are encountered again. Now let's learn about a B cell receptor. Each of these B cell receptors, they have membrane bound antibodies. Let's take the example of IgM antibody here. So this is a IgM molecule. Apart from these IgM molecules, the B cell receptor complex has two other important proteins called Ig beta, immunoglobulin beta, and immunoglobulin alpha. These are the ones which work somewhat like CD3 and zeta proteins, which we discussed in T cell receptor, right? So they have similar kind of function. They are the ones which help the B signal to. They are the ones. These are the ones which help the B cell to send the signal when it is activated, when the immunoglobulin binds to an antigen. Now, we need a minimum of two of these immunoglobulins for effective binding of antigen. Okay, only then these two can help to generate a signal for these B lymphocytes. When B cell receptor encounters an antigen with the help of Ig beta and Ig alpha, a signal is generated. That's a major signal. Apart from this major signal, there is a secondary signal as well. How does that happen? That happens with the help of another protein, another molecule called CD21. This molecule acts as a receptor for products of complement system, and that's a complement protein which binds to the antigen in question, right? So antigen binds to the complement protein, and this complement protein is identified by this CD21, thus generating a second signal. Of course, only the first signal is enough for B cell activation and response. If there is second signal, it's all the more good. Signal 2 may not be needed, but then if you generate lots and lots of complement proteins, these complement proteins, bind to the antigen and thus augment B cell activation and thereby B cell response. Let's move on to understand the dendritic cells. These are also referred to as interdigitating dendritic cells or interdigitating cells. These cells have a lot or elongated cytoplasmic processes. These are the antigen presenting cells for initiating T cell responses against various protein antigen. They are located at the right place to capture these antigens. What is that right place? First one being under the epithelia, which is the most common site of entry of microbes and foreign antigens, right? So in the skin, the immature dendritic cell is referred to as Langerhans cell. The second important site is interstitia of all tissues where antigens may be produced. So how do these dendritic cell act? They express lots and lots of receptors, and they also express a very high level of MHC molecules, MHC and other molecules, which help in capturing and responding to the microbes. And in response to microbes, these are recruited to the T cell zones of various lymphoid organs. Okay. In the T cell zones, these dendritic cells present those antigens to the naive T cells. And these naive T cells then they are activated and then they become effector T cells. There is another type of dendritic cell called follicular dendritics. They are called follicular cells because they are residing in the they reside in the lymphoid follicle, particularly in the germinal centers. They're found in spleen and lymph nodes. Remember the receptor for follicular dendritic cell, they don't capture antigens directly from the pathogens itself. Instead, they have receptors for FC, receptor for immunoglobulin G, and they have receptors for complement C3b. See, these receptors, they trap antigen which is bound to the antibodies, whereas receptors for C3b trap the antigen which are bound to complement proteins. Once the antigen is bound to these follicular dendritic cells, they present these antigens to the B cells. Remember, the regular dendritic cells are the ones which are antigen presenting cells for T cells, whereas the follicular dendritic cells, they present the antigens to the B lymphocytes, where the selection of B cells happen. The selection of B cells to mature into plasma and memory cells. How does this selection happen? It's only the best B cell is selected. What is that best B cell? The B cell which has antibody, the B cells which have receptors which are having very high affinity for these antigens. those are considered as best cells and they are selected. And they are the ones which go on to become plasma cells and they produce lots of antibodies and then they also go on to become memory B cells. Next important cells are the macrophages, which are again derived from the hematopoietic stem cells in the bone marrow in the postnatal life. These are the dominant cells in most of the chronic inflammatory reactions, and they are professional phagocytes. You all know in the peripheral circulation, it's called monocyte. Once it comes to the tissue, it's called macrophages or histiocytes. Why it is important? Because the half-life of monocytes is just 1 day, whereas the half-life of or the lifespan of macrophages may range from several months to various number of years. The role of these macrophages is twofold. One is they can present antigen to the T cells, resulting in activation of T cells. I mean, these antibodies help in induction of adaptive immunity. So these macrophages also, help in the effector phase of adaptive immunity. How does that happen? Because the T cells can activate macrophages where the phagocytic nature of these macrophages is enhanced. Okay, so they can either be inductor or inducer of adaptive immunity, or they can be effectors of adaptive immunity. Lastly, let's see what are all the tissues of immune system. These are primary lymphoid and secondary lymphoid organs. The primary lymphoid organs are the thymus and the bone marrow, where thymus is a place where the T cells develop, bone marrow is a place where B cells and other blood cells develop. Secondary lymphoid organs include lymph node, spleen, and cutaneous and mucosal lymphoid tissues. Lymph nodes, we all know that these are the nodular aggregates of lymphoid tissue along various lymphoid channels across the body. They respond to lymph-borne antigens. Whereas spleen, we all know it's an abdominal organ which responds to blood-borne antigens. Whereas cutaneous and mucosal lymphoid tissue, which are present or which are there in under the epithelium of skin and the gastrointestinal and respiratory tract epithelium. They respond to antigens that enter through various breach in these epithelium. Okay, so these are the various tissues of the immune system. So with this, we learned a bit about normal immune response in the that's about the concepts behind adaptive immunity. We talked about humoral versus cell mediated. We looked into lymphocyte diversity. We discussed in detail about the T cell receptors and other cells, right? So in the next session, I will explain you the concepts behind major histocompatibility complex molecule. We will see what is MHC1, what is MHC2, and what are the what is the clinical significance of understanding the concept behind MHC molecules. Thank you for watching. If you like this video, hit the like button. Do comment if you have any queries to ask. If you find this video useful, please do consider subscribing this channel and don't forget to share. Thank you.